taken if it's controlled by one's will. Go on, try to subtract. Maybe Shafika. Question 1, yeah, answer. STQR. Yeah? Okay, STQR. So where does S start from? Starts from the brain, then it goes to T, then it goes to Q, then it goes to R, and R here being the muscle. Okay? How do you tell that this is a motor neuron and not this? Because no arrows were shown, right? So you don't know the direction of flow of nerve impulse. So what are the clues? How do you tell this is motor and not sensory? How do you tell this is sensory and not motor? What are the clues? Thank you, Henry. For the sensory neuron, body is in the middle. That's the most obvious clue. Once you rule that out, you will know what this one has to be. This one is likely the motor neuron. Thank you. Some of you may just realize, you see, you start from the brain, S, then go to T, then still go to Q. What might these neurons be? Okay, it is highly possible that you have multiple relay neurons before you get to a motor neuron. Okay, what we provide you is a very simplified neural circuit. Sensory, relay, motor. But the relay, I could do away with it. I could have 1, I could have 2, 3, 4. Really depends on sometimes the distance, how much processing needs to happen in between before it reaches the motor. Okay, so be flexible in that sense that we can have more than one relay neuron. If we are just looking for a relay reflex arc, then I think you can choose P, Q, R. Yeah, if we just want a reflex. P, Q, R. Go in and then come out. Next question asks you to define what a reflex arc is. Okay, they say what is a reflex arc? Part 2, they ask you to describe the reflex action when the finger touches the sharp end of the nail. Yeah, I like to say that usually such questions, the response is very standard. Only thing that could change is we change what the reflex might be. Other than that, there are very standard steps to describe a reflex arc. First and foremost, what's the reflex arc? You can define it as such. Shortest route the nerve impulse will take. From where to where? From a receptor to the effector. Okay, that's what a reflex arc should go by. For something that's voluntary, just start from the brain or do. The second part is the one that expects you to write a little bit more. This is a 5-mark question. You can see how we roughly allocate the marks. Always, the start should start off with some sort of a receptor. We may not tell you what the receptor is, but you have to say some sort of receptor. Let's say if you are seeing something, logic will dictate that the receptor detects light. If you feel pain, the receptor detects touch. Yes? Yeah, if you do not know what kind of receptor it is, you don't know what's the name of it, you can just say receptor that detects, then you fill in the blank. Okay? Fill in the blanks of what the stimulus is. Is it light? Is it pain? Is it the change in temperature? Right? If you don't know the name of the receptor, it's fine. Just receptor detects whatever the stimulus is. Oh yeah?
Is detecting and stimulating.
Ah, okay. Detecting and stimulating is, it could be one and the same thing. Because when a receptor detects something, a stimulus, it will then also go on to stimulate and create a nerve impulse that is sent to the sensory neuron. I think if you want to use the word stimulate, you can use it towards the end. You look at the first sentence, sharp end of the nail is detected by pain receptors. And this stimulates the production of nerve impulses. If you want to use the word stimulate.
Can you say like the sharp end of the nail stimulates the nerve?
Okay, I have seen people write that. I think it's accepted. You could also say the sharp end of the nail stimulates the pain receptors to produce a nerve impulse that travels then next to the sensory neuron. The ones in between are familiar to you. Mention of sensory neuron going to the central nervous system. Then there will be some sort of a relay neuron that will then relay this to the motor neuron. The very last step of such a question usually ends off with what the effector is. So from the motor neuron, you transmit the nerve impulses to the effector. In this case, it's the muscle to withdraw the finger. So do you know what we can really change? The main thing we can change is the receptor and the effector. Because sensory, relay, motor neuron, we do not differentiate it to the types very specifically at your level. Right? When you go to university, perhaps, in the eye, we may call that relay neuron, that motor neuron, that sensory neuron a different name. At the foot area, we may call it a different name. But for us, we just generalize these three types of neurons. So really, we can change the context, but the middle part will remain the same. The receptor part is the one you may want to change. What are you detecting this time? Is it light? Is it change in temperature? Is it pain? Last one, what is the effect? Do you pull your hand away? Do you blink? Do you pull your leg away? Do you kick out? The effect may be different. So that is where you change the context. But you'll find that it's largely very similar. So let's look at some of your friends' responses. Okay, we look at the first one. When the finger touches the sharp end of the nail, sensory neuron will detect the touch and transmit nerve impulse to the central nervous system. Then motor neuron transmit nerve impulse central nervous system to the effector. What is missing here? Relay neurons, right? Notice there's no relay neuron here. Look at the context. If they show you a neural circuit that has relay neuron, that's something you want to put in. Next, there's no receptor. The sensory neuron may not be the one that is the receptor. Okay? I'll just put it up front to you. In biology, some sensory neurons act as receptors too. Okay? But not always the case. So for us, we play it safe. You always say that there's a receptor, then a sensory neuron. But if you study biology in university, you'll find that some sensory neurons act as receptors too. Look at the second one. Sensory neuron detects the sharp end of the nail as a stimulus, then connects with a relay neuron, which in turn connects with motor neuron. This friend of yours mentioned connects. But so what if it connects? It needs to transmit. Okay? It may be connecting, but if there's nothing transmitting, then actually no information is flowing. Same here. The starting point here is the sensory neuron. We need a receptor. Okay, we need something to detect the stimulus before you can pass it to the sensory neuron. Let's look at the next one. Your friend over here mentions as soon as our finger touches the nail, receptors on our fingers will send nerve impulses through the sensory neuron. I think this is okay. Starts off from the receptors, then the nerve impulse generated, it'll pass the sensory, and then it'll pass the relay to the spinal cord, into the motor, then to the muscles, which is the effectors. This is a very complete answer. Okay? So for yourself, you have a look. I think likely the things that you may miss out will be the receptor and the effector put in context. And did you mention the flow of the nerve impulses, or it's just a connection between them? Okay, we look at this particular answer here. You see that the sensory neuron connects to the relay, which connects to the motor. Nerve impulse are then transmitted to the muscles in the arm to withdraw. This one almost implies that nerve impulse only flows through the motor neuron, and then it skips flowing through the sensory and relay. Yeah, we look at the option at the very top. Okay, so you want to mention that nerve impulses are transmitted from one neuron to another. Okay, so have a look at your responses. This one, although it's just in bullet points, you shouldn't write in bullet points in your actual answer, but very succinct. It does cover the key things that you should at least put in. from receptors to sensory, the central nervous system, then the relay. Okay, this person copied from textbook and paste there. Yeah, but these are the key things that should be in your essay. But you write it out in prose. As I said, the receptor could be different and effector could be different depending on what reflex you're talking about. Yes, Harry. I would suggest that you look at the case study provided. Yeah, the case study that we provide you, we could show you that the relay neuron is inside the spinal cord or the brain. So you look at the diagram. Okay. Last but not least, this is another. Okay, they ask you to state two ways that a reflex action could be different from a voluntary action. Let's look at some of your responses. First one, reflex action. Relay neuron may not be used unlike in a voluntary action. The pathway of nerve impulse starts from the forebrain for voluntary, or it starts from receptors for reflex. I think that's okay. Next one, some reflex do not involve the brain or relay neuron in the process. That's fair. Or voluntary actions do. Voluntary actions should involve the brain. Good. Reflex action requires a stimulus, while voluntary action does not. I would agree with you, but perhaps don't make it too absolute. You say a voluntary action does not. I think this is iffy, because just like how you catch a ruler, technically there was a stimulus of some sort, but you decided whether you want to catch or not. Okay? So rather than say, well, voluntary action do not, a voluntary action may or may not require a stimulus. That keeps things a bit more open-ended. As we scroll down, we see that a lot of your responses are roughly similar. A big difference you all mentioned is the source. Where does it start from? Voluntary, the brain. Involuntary, you really need a stimulus of some sort. Okay, this particular option here, voluntary action is deliberate. Involuntary action is a reflex action. I suggest not to write down such a comparison, because the name of the action itself already implies that it is voluntary or involuntary. Yeah? Voluntary action is deliberate. I could have just said voluntary action is voluntary action. Yeah? So we try not to describe the name. The second one is okay. Voluntary actions do not require a sensory neuron. It may or may not require, while involuntary actions require a sensory neuron. A neuron. Okay, so largely I see that around the same kind of responses. Oh, this person put it in a table, which is okay. Wow, goodness. Okay, I want to compliment this person. Who's this? Kyra. Okay, Kyra put it in a table. Okay, but in a table, perhaps now you can see clearly whether we have got all the differences. Okay, voluntary. Relay neuron, do we require it? I would say most of the time require. Reflex sometimes. Presence of receptor. Okay, may or may not. Reflex present. Sensory neuron involved. In the main circuit, perhaps not. Reflex all the time. Starting point, the brain, receptors, nervous system. Okay, this one somatic nervous system can involve reflexes. Okay, please take note. This is a common misconception. Somatic nervous system, it can be voluntary. It can also be of reflex nature, involuntary. And perhaps the best example I can give you is, you can voluntarily control your arm. Oh goodness, okay. Okay, look at chicken wing by this arm. You can voluntarily control your arm to grab things, right? I also don't know why I drive. A reflex action, if you touch something hot, you also automatically pull back, right? Okay? So somatic nervous system can be voluntary or involuntary. The main thing is that they control the skeletal. So it's a misconception to think somatic only controls the voluntary and it ignores the involuntary. That's not true. Okay, it includes the reflexes. Autonomic though, autonomic is purely involuntary. Okay, but this one both, voluntary and involuntary. Okay, I think maybe this is around the third time already that we have surfaced this mis- Wait, what are you playing? Okay, muscles involved, correct? Skeletal? Non-skeletal or skeletal? Okay, yeah, fair enough. Some reflexes may not involve skeletal muscles. Okay? Now, let's close off the chapter. If you have any interesting questions you'd like to ask to close off the neural circuit section, after that, we'll slowly dive into learning about the eyeball, one of your most major sensory organs of the body. Next time I see you on Thursday is where we will look at real one. Okay, we'll take a short pause for you to consolidate your learning, write down some points, anything new you learned, and I'll answer some questions.
No, it fell on me, so it's good.
Okay, yeah. Okay, for you, it's okay. Your chest muscle quite solid, right?
Yeah.
It's okay, I really want to change the iPad. So if you break it, I have a good reason. Yeah. And then you can help sponsor it. How far you know why I chose you?
Why? It's just a break.
Then we break, right? Then I can how far you go eat and make some money. Okay. Any questions on the ground? Then I see your full name, I get confused. Ella, what are the case studies we need to know for this topic, especially if it's essay questions? There are two case studies of neural circuits for reflexes, right? The two case studies are the withdrawal reflex, number one. Number two is the knee-jerk reflex. These two case studies we expect you to know. But truthfully, the main difference between these two is the presence of the relay neuron or not. Okay? If we expect you to know these two case studies, but we provide you new ones, it will not be too out of this world and far-fetched. It will be quite doable. Para, yes. Wow. Okay, Shafika asks, it's a very good question. Is the effector always a muscle? Not necessarily. There are other effectors. For example, glands. Your salivary glands are effectors. When I see something tasty, my salivary glands squirt out the saliva. Yeah? Have you experienced it yet after I told you about that? Yeah, sure. Yeah, the mood. Go and try really, like you become like a viper. You must find something that you really find tasty. Then your mouth must be very dry before it happens. Then you look at that thing and okay, what exactly is the receptor like? Okay. I deliberately left this out because it's complicated. What is a receptor? I like that you asked this question. Now that you know what biomolecules are, right? So always ask yourself, what is this thing we are teaching you? Okay. Since the question has been asked, I'm going to tell you that a receptor could be multiple things. Some receptors are just the end of the sensory neuron. Some receptors are specialized cells that purely just detect the stimulus. And then a sensory neuron is after this. Okay? Okay, so what's my reply to Shabika's question? What are receptors? They could be the... Where is it? A receptor could be the end of a sensory neuron. Or it could be a specialized, how do you spell special? Okay, receptors could be two things. It could be a specialized cell, or it could be the end of a sensory neuron. These two things here, they are both sensory neurons. In your eyeballs, there are specialized cells that detect light. After they detect the light, then they pass the information on to sensory neurons. But some sensory neurons act as receptors by themselves. Apa ya? Question?
Oh, I was just saying how when I fell, then my eyes detect. Then like my brain send the thing to my hand muscle that I connect. Yeah.
So, do you think that's a reflex?
That's not reflex.
Okay, you talk very fast. Your reaction time very fast.
Yeah, I mean just now, I mean yeah.
Yeah. Good job. I'm glad your reaction time is fast.
Yeah.
Can reflex actions use more than one part of the body? Oh, okay. That's a good question. I don't know if a reflex action often just includes just one circuit to control one effector. I don't know. But I can imagine how one stimulus can stimulate multiple different reflexes at one time. For example, for you to be able to see things clearly, I think multiple reflex actions are occurring to control various parts of the eye so that you can see clearly. Okay? So I think one reflex action for one action, but one stimulus to stimulate multiple reflex actions at one time. Okay, show new cards, any more new questions? Okay? Is screaming when you get scared also a reflex action? I don't know. Siqi, please tell us Google now. I'm very curious. I've not screamed before when I get scared. So, I don't know. Do you normally scream when you get scared? Alpha, do you scream when I pep down? Oh, okay, maybe I scream, but then in my mind, it's not considered a scream. For me, when something scary happens, I'll be like, yeah, that's as far as it goes. It's not like a. Huh? Siqi? It is, It is, okay. Okay, then the next question is what might be the biological function, purpose of screaming? Okay, perhaps it's to attract attention of the predator. Okay, maybe to get people up. Maybe. Yeah, reflex action usually have a biological purpose for your own protection, right? Okay, Juni asks, how to phrase? Can we use somatic and autonomic as an adjective? What's an adjective? To describe a what? A verb. Okay, this action was somatic. Oh, okay. Usually I see people use it this way. Somatic is normally attached with the one nervous system. So someone may say that this particular action is controlled by the somatic nervous system. Yeah, usually they'll say it's controlled by which nervous system, somatic or autonomic. Somatic, you don't attach it to usually they attach it to nervous system. Okay, this is Shriya. Do we call the impulse of nerve impulses? Okay. Call it nerve impulses. Okay. Are you wrong to say electrical impulse? I see a lot of sources say, but I feel that when we say it at our level, we don't fully appreciate what it means when you say electrical. Okay, so we say nerve impulse will do, so that people don't question you too deep. Is there a specific order to the sensory, motor, and relay neurons? What do you mean by that? Okay. No. So a motor neuron's function is really to get the effector to do something. It cannot be the one to detect a stimulus. Okay, that's the role of the receptor or the sensory neuron. Okay, because Shafika asked earlier on, what is a receptor? And my reply is that receptor could be the ending of the sensory neuron, or it could be a specialized cell that after being stimulated, pass it on to the sensory neuron. Okay? Any more questions? Okay, nil. Okay? And that is all. We'll close off this part of the chapter. So this is what we've covered so far. Every system, we start off with the parts of the system. We've learned about the 3 kinds of neurons. We learned about, in this section B, how they work together. Now in this last section, we're going to look at a case study. We're going to look at how your eyeballs work. But before we look at how the eyeball works, we need to first know what is the structure of the eyeball. Yeah? Structure first, then we know how the parts work together. Today, the purpose of today's lesson, the last part, we're just going to roughly introduce you to the parts of the eyeball. Next lesson, then we look for the real parts when I dissect it open. After today's experience, my best advice to you is, I encourage you to come close to me when I dissect, but not too close and put your face right in front. Because when I poke into the eyeball, it will spurt. Okay, and today I have a few students came too close. So I don't know, was like, poke and after that, okay? And the liquid inside that will come out, right? It will look black in color. Okay? The liquid is not actually black, but because of one layer that I cut through, that releases all the black pigment. After all, you notice when you look into someone's eye, the pupil, that circle where light goes in, notice there's black color, not like any other color. Yeah, it's black because of the pigment inside your eye. Okay? So today, I'm going to introduce you very broadly to the structure of the eyeball before we do anything else. Okay, the idea is to go to SLS, you can click on virtual model of the eyeball, and this is what you will get. Okay, please have this on standby so that you can move along with me. Let me introduce you to the parts of the eye. I hope I don't need to tell you that this is just half an eyeball. Yeah? Okay, okay. Sometimes I need to say clearly because I realize over the years when I teach anatomy, usually diagrams, virtual models, they like to show you a dissection of it. But some students actually think that it just looks like that. Okay? Your eyeball is fully round, huh? It's not just like that. Okay, so if I were to slice an eyeball down this way, this is what you see on the inside. During the practical this Thursday, I will not slice it this way. Instead, I will slice it, okay, from top down. I'm going to slice it again. I'm going to slice it again. Okay, instead. You can see a lot more structures by doing it that way. Okay, so today I just introduced you to the various parts of the eyeball. Your task is, you see at the very bottom, there's a drag and drop where you fill in the blanks. Yeah? You see a drag and drop? Okay, if you're able to do that and accomplish that by the end of this lesson, I would say that you know largely most parts of the eyeball. Okay, let's begin our lesson. First and foremost, when you look at any eyeball, one of the most common things that you think of is that the eyeball is white in color. Okay? Yeah, that's because the bulk of the eyeball is white in color. Look at each other's eyes, right? White color? White color, yeah? Okay, that's the side of your eyeball. We call this outer coating the sclera. S-C-L-E-R-A. Sclera. If I have a daughter, maybe I'll call her sclera. Sounds like a name. Yeah? Sclera Tan. If I'm not wrong, scleras can come in other colors. Okay. Yeah. What? They tattoo their eye? Oh, goodness. Okay, I don't quite see other colors. But how you ask, when people tattoo eyeballs, where do they tattoo? Actually, they are tattooing the sclera. You see, so white, right? That's the only part I could possibly color. Yeah? Then you may be thinking, hey, there's some people iris, right? This part over here, different color one. Yours also a different color. Okay, what do? They wear contacts, special contacts to make it colored. We do not tattoo the iris, okay? Cannot tattoo the iris because the iris is just a bunch of muscles. that muscle controls the window into your eye, how big and small this pupil will be. Okay, this hole into your eyeball. This is the sclera that you can tattoo. But I also don't know how people can keep their eyes open while they're being tattooed. Okay, let me search in the other colors. Animal. Okay, some chimpanzees their sclera is a different color. You look at this chimpanzee here. You can see that its iris is kind of brown in color. You look at the sclera outside, it's blackish. Yeah? So, human eyeballs, our sclera is white.
Wait,
Yes?
is our iris like exposed or is there something covering?
Okay, good question. Okay, so let's go to the next structure. What is the function of this white coat? Honestly, this structure here is very, very, very tough. Today, when I was trying to pierce through the sclera, I had to use my whole, almost like my body weight, right, to puncture in. And then the moment I puncture in, right, and boom, all the liquid come out. Okay? So, the white structure here, this is the sclera, is very thick and very tough. Right? Today I forgot to bounce the eyeball. But actually, it's that tough. You can bounce. Yeah. Some years, when it's round enough, I just drop right to the point. Okay? Okay, next. Your eyeballs come with muscles attached to them. There's one on top, one below, one on the right, one on the left. And so this is how your eyes work up. If I want to look this way, this muscle will contract and this muscle will relax. So contract and it pulls. Then I want to look the other way, this muscle will contract while this one relax and lengthens. If I look up, same idea. So that's how you look left, right. And you want to look all around, you can try it now. It's not that easy to control your eyes look. Okay? So the muscles are there for you to control the directionality of your eyeball. Now let's start to look at the inner parts. We often say that can you see this black part over here? Yeah? That is your pupil. Okay. I don't know if you've actually seen your own pupil before. If you don't feel shy, you don't mind, you go and look into your friend's eye. You see if you can see the pupil. It is different from the iris. A lot of our iris is also very dark in color. And then, but if you look very carefully, in the middle of the iris, there is one very dark black circle. Okay, that is the pupil. Can see? Can see? Okay, cannot see, then I'll show you mine. Okay, this one I got my brother to help me take. Yeah. Okay, so this white part is my sclera. This part is my iris. I want you to look at my pupil. There is a difference. It's just that sometimes the color is so similar that you just think you have one big black hole there. Okay, it's not, there's a small black hole. Can see my pupil? Yeah? It was on this day after many years that I realized that my iris is not black. My iris is actually brownish in color. Yeah? Do not try this at home. I shine one bright torchlight right into my eyeball. Yeah? Because I want to see my pupil change size. You saw my pupil change size? Yeah. So this is when my brother shined the torchlight. Saw the pupil? Yeah? Okay, in the name of science, that was quite painful. Okay, one more time. Boom. Yeah? Do not do that. When you do that, the light goes straight to your receptors and it can scorch and burn your receptors until they cannot detect light. Ever stand into the sun before? Yeah. And then after that, you look away, you close your eyes, you see one bright patch there? Yeah, that bright patch, right, is the receptors in that region that have been scorched and burned. Okay? So don't do that to your eyeballs. Okay? Yes? Okay, when I say scorched, right, means, right, at that very moment they're traumatized. Okay? Too much sensory stimuli. But you do it for too long, you can damage it. Momentarily, it was just an overload of information. Yes? Okay, in the past, right, neuroscientists used to think nerve cells, right, cannot divide once they've specialized in the nerve cells. And so people are very worried when you hurt your head, right? One of the arguments was that once you lose neurons in your head, right, it's gone forever. Yeah? But new studies are showing that once you lose neurons, it's still possible for it to respawn and create new neurons. Yeah? Okay, respawn not in a game way, I know your game, your respawn is like come out of nowhere, fall from the heavens. Okay? Okay, the next thing, okay, just now, Alpha asked, your pupil and your iris, actually, can you directly touch it? You can't. Have you ever tried touching? Okay. What you are touching there is not your iris directly, it's not your pupil. You are actually touching something called the cornea. So you look at your virtual model. If you look at your virtual model at the side view, can you see that there's one transparent layer popping out? That is the cornea. And if I look from the front view, you can see your iris. That's because your cornea is transparent. Okay? It looks like there's nothing there, but there is something. Okay, there's a transparent window. Underneath the window, then that's where your iris is. The cornea is actually connected to the sclera. So the eyeball is like that, mostly opaque and white, but at this juncture onwards, it becomes transparent. Okay? I say again, the eyeball is mostly opaque and white, no light can go in elsewhere. But at this juncture over here, it becomes transparent. Alright, and that transparent layer is the cornea. And this is actually the first point through which light can enter your eyeballs. And that is where light bends the most. Because behind the cornea is a lot of liquid. Liquid that will burst out, right, when you poke into it. There's no liquid in this, don't worry. Yes, Rafika? I think if you can absorb light through the rest of your eye, then you will not have the ability to be able to concentrate an image onto the sensory, to the receptors of the eyeball. Okay, the light that will, if light can come in from anywhere, I think it will be too scattered for you to properly focus an image. So that's why it needs to come into a focus point. Okay, so here, if I'm going to remove more parts, the cornea is a transparent layer. This is an inaccurate model because by right we cannot just remove the cornea like that. Okay, it is part of the, it is fused to the sclera. But behind the cornea, there's an empty space. Yeah? It's not really empty, it is filled with liquid. In your textbook, they call it aqueous humor. Aqueous meaning like aqua, water. Okay, so it's very watery fluid behind your cornea. What is it for? Well, it helps to refract light. You know when light changes medium from air to water, it bends, right? You all learn this in physics? Okay? When light passes through from air to liquid, it will bend. I'm going to remove the cornea now so that you can see something. Yes, Kara? Of the what? Yes. Okay, you need to know the name of the liquid, aqueous humor. I have now removed the cornea. And after I remove the cornea, now you see, I can actually, there's a hole. This is where your pupil is. Your pupil is not a black dot on your eyeball. It is literally a window into all the way to the back of your eye. So when you look into someone's pupil, you are looking straight to the back of your eyeball. That's what you are doing. Yeah? I think it's quite romantic. Yeah? No man? I mean, that's what I tell my wife. I'll be like, I'm not just looking into your eyeball, I'm looking to your back of your eyeball. Yeah? That means the back of your eyeball is blackish also lah. Okay, what you are seeing is the black at the back. We can control the size of this hole that we call the pupil. Yeah? And we control the size using these muscles surrounding it. This part that gives your eye the color actually is muscle. We can use these muscles to make this hole smaller or bigger. Okay? But I need to say, as small as you can get, you can never close this fully shut. So let's say if there's too much light coming to your eye, and your muscles have already tried to shrink the pupil as much as possible, like my eye over there, what's the next best thing your eye can do? Yeah, that's what your eyelids are for. Okay? If there's really too much light, the last contingency plan is your whole eye will just stimulate the blink reflex, and you're just like, okay, that's what you do. But when you close your eyes, actually your pupil is still open. What is the bulk of your eyeball filled with? It is filled with another substance. In your textbook it says vitreous humor. This particular substance here is jelly-like, not like water. Okay? It is jelly-like. But both the aqueous humor and vitreous humor, they are both transparent, so they light to pass through. Then you may be wondering, how come when I slice the eyeball, it comes out as black liquid? Okay, that's because if you look at this eyeball here, you see a dark layer here. Yeah? This dark layer inside this model over here, I flip around. This dark layer in this model is represented by the pink layer. You see the pink layer? Yeah? This layer here, this pink one, is called the choroid. C-H-O-R-O-I-D. The choroid layer contains pigment. It also contains a lot of blood vessels to transport nutrients to the eyeball. So, when I slice into the eyeball, I inadvertently slice through the sclera, and I will break into the choroid, which is filled with black pigment, and the black pigment will spill into the liquid here, and that's why when the liquid comes out, it comes out black. But actually the liquid is transparent. Yes? Okay, what's the pigment for? Okay, I'd like you to think about, why do you think the choroid has a very dark black pigment, as opposed to not have pigment at all? Okay, yeah, it really has to do with light. Yeah, I don't know if you learned before, but in physics, dark things have absorbed light better. So that's what we would want. We don't want the light, we don't want the inside of the eyeball is to be fully reflective. Then the light come in, it just ping pong, ping pong, ping pong, ping pong, right? And you never really receive any information. Right? Best part, after it goes in, right, then reflect out of your own eyeballs. Yeah? Then you're not going to detect the light. So we want it to stay within your eye. Some animals, however, they have a layer, just one thin layer at the back of their eye that is reflective. You tend to find that animals that tend to roam around in the dark. So when it's too dark, you want whatever light, even very little that gets into your eye, to stay inside. And so these night-living animals, they tend to have a small reflective layer. That's usually how you can tell. Yes? Why must your eyes be wet? Like which part wet? Like inside wet or like outside?
Like you blink with the moisture in your eyes.
Oh, okay. You blink is to moisturize your eyes. I think it is for lubrication. Can you imagine without tears, without lubricant, you every day blink your eye how many times? You're going to get eye abrasion, I tell you. Yeah? And that's why when your eyes are a bit too dry, and if you blink, it gets very uncomfortable. You feel this burning sensation. Yeah? So you need it to be lubricated, so that when you blink, it's not painful. It is like, I ask you, take off and put on your clothes many times every day. You will confirm get some abrasion in your armpits after a while. Yeah? No? Okay, sorry. How do you take off? Where will you get abrasion? Yeah, I don't know. Okay? Any other structures of the eye I'm going to share? One last thing. This Thursday, when I show you the structure of the eyeball, you'll find that there's one tube coming out of the eye, at the back of it. Remember I said your eye sockets got two more holes at the back, so they can connect to the brain? Yeah, that is for this tube that's coming out of the eye. This is where the nerves will come out from, the sensory neurons and the motor neurons can go back in via. Our blood vessels also go in and out via this tube. On the inside, you will see an opening, usually, where all of these will branch out. Okay? I think I've covered quite a lot of structures of the eye. Okay, in this remaining time, I'm going to show you just a few more. Some of these structures you cannot see even when I dissect it. The best option is to look at virtual models. First one, you look at number 4. Number 4 is a lens that has been cut in half. So every eyeball comes with a lens. Every eyeball comes with a lens. This lens on Thursday can take out one. That's the thing I will take out on Thursday and I'll show all of you. Actually looks really like that. The lens is not like this lens, it is not solid. Okay? It's not like your physics, you all use those mirrors. The lens we have is actually squishy. It is like a water balloon. Okay? But the beauty of having a lens that's like a water balloon means that I can control the thickness. The thicker the lens is, the more light bends. And if I stretch the lens on its edges and make it thinner, it bends less light. And that's how our eyes control how much light bends. It controls the thickness by bending it. So our lens is not solid. Yes? Yeah. I mean, you try right now, you put your hand up close, okay, it's clear. You bring it far away, it is still clear. But for you to achieve that, it comes down to your lens changing its shape from thin to thick, thin to thick. The next interesting thing is, the way we change the shape of the lens is not quite what you imagine. The way we change the shape of the lens is using small little strings attached to the edge. So if you look at this model of the eye, can you see that at the edge of the lens, there are many of these string-like structures? If I want the lens to be thinner, these string-like structures will then become very tight, and the lens will become much thinner. If I want it to be thicker, these strings will become looser, and then the lens will become thick. We'll learn more about this in subsequent lessons. Here today we just know about the structure. We call those strings suspensory ligament because it suspends the lens within the eyeball, and it's a ligament. Okay, remaining part of this lesson, may I get you now to look at the diagram provided in the last part of the SLS? May I get you to try your best to drag and drop to match the parts of the eyeball? Next lesson, when I see you in the lab, then you will appreciate the structures that you see in real life a little bit better. Last lesson. Sorry, last lesson. Okay, before I go up, I want to show you something interesting. You know your pupil is round, right? Not all animals' pupil is round. Have you ever seen the pupil of a goat or a sheep? Yeah? Yeah, so not tomorrow, Thursday, the sheep eye, the pupil looks like that. So you may be thinking, hey, how come the eyeball deformed one? No, it's not deformed, it really is like that. Okay, the pupil is this shape. I think frogs, let's look at frog. Pupil of animals, how about that? Then you can see the variety. Okay? You see the octopus one? Yeah? Look at the cat's one. It's rather sharp looking. Okay, ours is quite unique. Ours is round. Hey, wow, it's fake, right? Oh, gotta be fake, man. Okay, so many different kinds of pupils. Right? All of us evolve different kinds of pupils. Alright, that's all for today. Next time I see you, we will look at a real one. And then, I will say, if you do not feel comfortable, please don't come too close. Just sit far away. I will play on the big screen. You can watch from afar. Okay, that's all. Junaid, help me collect, and then you can put my picture. Okay, thank you, class.